Auzubillahiminash-shaytanir-rajim. Bismillahir-Rahmanir-Rahim. Hope you people are fine. The topic that we are going to discuss today is transport through the cell membrane, specifically osmosis. The learning objectives for this lecture are that at the end of this lecture, you will be able to define osmosis, you will be able to define the osmotic pressure, and you will be able to describe the effect of tonicity on the cell volume. As you know that the cell is surrounded by a cell membrane, and the fluid which is inside the cell is called intercellular fluid, and the fluid that surrounds the cell is called the extracellular fluid. And anything that has to pass between the cell and the surrounding extracellular fluid must be able to pass through the cell membrane. It forms a boundary for the cell. The cell is surrounded by a phospholipid bilayer, which serves as a barrier around the cell, especially for the water-soluble molecules, because it is a phospholipid layer. However, for the cells to survive, some of the materials they need to be able to enter and leave the cell. All the material inside the cell cannot move out, and the material outside, the solutes, all the solutes, everything cannot pass through the cell membrane, as it is a selectively permeable membrane. And make a note of the point that there is a difference in the composition of intercellular fluid and extracellular fluid. Like, for example, in the cell cytosol, there are large proteins, organic molecules. They stay in the cell, they cannot leave the cell, and there are certain phosphate molecules and some other larger ions which cannot leave the cell. And similarly, all the solutes, they cannot pass, most of the solutes, they do not pass the barrier, and this difference in the composition of the intercellular fluid and the extracellular fluid has to be maintained. But to maintain the osmotic equilibrium between the two compartments, it is the water which can freely move through the cell membrane. So let us have an overview of the different mechanisms by which the transport occurs through the cell membrane. One is the simple or passive transport, the other one is active transport. Passive transport means the transport that occurs without the utilization of energy. The energy is in the form of ATP, and the mechanisms which are simple transport mechanisms which do not require energy are diffusion, facilitated diffusion, and osmosis, while active transport includes those mechanisms which require energy in the form of ATP, like primary active transport and secondary active transport. Today, the basic mechanism that we are going to discuss today is osmosis, which is a passive transport mechanism that does not require energy. So let's see what is osmosis. Osmosis, in simple words, it means diffusion of water across a semi-permeable membrane. The plasma membrane is permeable to water but not to the solute. And what is a solute? Solute, this you have studied in chemistry, and you know from your previous knowledge that solute is a dissolved particle, and the solvent is the medium, usually liquid medium, in which the particles may be dissolved. And if the solute does not readily cross a plasma membrane, it is called an osmotically active solute. And to define it specifically, it is defined as the net flow of the water molecules across a semi-permeable membrane from the compartment with lower solute concentration towards a compartment with higher solute concentration until an equilibrium is reached. So the water always moves from an area of low solute concentration to an area of higher solute concentration. And if we talk in terms of the solvent molecules, it is the net diffusion of the water down its own concentration gradient, that is from an area of higher water potential to an area of lower water potential or concentration. But usually when we discuss or when we describe osmosis, we usually give the reference of solute particles. Let's see what is the basic difference between the diffusion and osmosis. In case of diffusion, it is a movement of the solute particles from an area of higher concentration to an area of low concentration. But when we are talking of osmosis, we are not talking of the solutes. We mean movement of water. Please remember, movement of water across a semi-permeable membrane from an area of low solute concentration to an area of higher solute concentration. Let us take an example. If we, for example, if we have a U-tube glass and we have equal level on both the sides of water, and then if we add the solute, more solute particles on this side, the water will follow the solute particles and the level of water in this limb will start falling and the level of water in this limb will rise. This is because of the net diffusion or the net movement of the solvent molecules, that is the water, from the area of lesser solute concentration to an area of higher solute concentration. So the level of water in this limb will rise. This is due to, this rise of water is due to the osmosis. So let's take up another term, that is osmotic pressure. What exactly osmotic pressure means? It is the exact amount of pressure which is required to stop the osmosis is called osmotic pressure. And the osmotic pressure is directly proportional to the number of osmotically active particles. If, for example, we have this U-shaped tube and we have water in this pure water and we have a non-diffusible solute on this side, what will happen? The water will move through this membrane, semi-permeable membrane, towards the area of the solute concentration, and the level of water in this limb will rise. But if we don't want this to rise, what we have to do is to apply a pressure over here at the surface, top surface over here. And if we apply a pressure that is exactly required to stop the osmosis to occur, this pressure would be called as the osmotic pressure. The exact pressure required to stop the osmosis is called the osmotic pressure. And how much pressure will be required over here? It depends upon the number of osmotically active particles which are present over here. If there are a large number of solutes over here, a large amount of pressure has to be applied to stop the osmosis. And let's take up some important terms, some important definitions which are used in relation to osmosis and to describe. These are the basically concentration terms. Osmole is defined as the number of particles in 1 mole of undissociated solute. is called 1 osmol. This is your FSC knowledge. You know that 1 osmol has 6.02 into 10 raised power 23 particles. That is Avogadro number of particles. And osmolality, students should know the difference between the terms osmolality and osmolarity. Osmolality means the number of osmoles of solute per kilogram of water, while osmolarity means osmol per liter of the solution. And in usually the term that you'll be using more in physiology is osmolarity, because when we are talking of the body fluids, we usually refer to liter of the solution, and we talk in terms of the volume. So mostly the term osmolarity is used. So now you know that osmol is a concentration term that is used to express the concentration of solution in terms of number of particles, that is, and its unit is osmol. 1 osmol is equal to 1 gram molecular weight of undissolved solute. In 180 gram of glucose, it means it is 1 gram molecular weight of glucose. And if we talk in terms of osmolarity, osmoles, it is 1 osmol. But when we take, if we dissolve NaCl in the water, it dissociates into 2 ions, sodium and chloride. So 58.1 mole of NaCl, 58.5 gram NaCl will be equal to 2 osmoles and not 1, because it has 2 osmotically active particles. It dissociates. And when we say milliosmol, we mean 1/1000 of an osmol. And the term that, because we'll be dealing with smaller values, we'll be talking mostly of milliosmol. So again, to repeat, the osmolarity, when we talk of osmolarity, we refer to a solution that has 1 osmol of the solute dissolved in each liter of the solution. And when we say osmolality, we mean the solution has 1 osmol of the solute dissolved in each kilogram of the solution. And there is only very less than 1% difference between osmolarity and osmolality. They can be used interchangeably. And what is the relation of osmolarity to the osmotic pressure? At normal body temperature, 1 osmol per kg will exert a pressure of 19,300 millimeter of mercury. And if we talk of 1 milliosmol per kg, it exerts a pressure of 19.3 millimeters of mercury. And the normal osmolarity of the body fluids, of the both extracellular fluid compartment and the intercellular fluid compartment, is 300 milliosmol per kg. So to get the osmotic pressure, we multiply 300 milliosmol per kg times the pressure which it will exert. 19.3 multiplied by 300 gives a value of 5, to be very exact, 5790 millimeters of mercury. And if we round it, we take in a round figure, we will say it exerts a pressure 1 milliosmol per kg will exert a pressure of 5500 millimeters of mercury. Let's take up a new term, that is tonicity, and you are familiar with it. This is how the solution's osmolarity will affect the cell volume. What will be the effect of the solution in which the cells are placed on the cell volume? When we use the word isotonic, we mean the solution, which means that this solution has the same solute concentration as that of the cell which is placed inside. And when we talk of RBC, the RBCs, the same concentration of solutes in the RBC and the same concentration in the solution. So when this is the situation, equal amount of water will leave the cell, and same amount of water, same volume of water will enter into the cell. So there is no net change in the cell volume. And when we say hypertonic, we mean solutions having greater solute concentration than that of the cytosol. The water leaves the cell and the cell shrinks. As you can see in the middle here, a speculated appearance because of the cell shrinkage. And hypertonic means the solutions having lesser solute concentration than that of the cytosol. In this case, the water will enter the cell and will cause the cell to swell, and there is a chance that the cell will lyse. If there is too much of water entering into the cell can burst or lyse. Here you can see the situation when the cell is placed in a concentrated salt solution. The water moves out of the cell and the cell shrinks. This is called plasmolysis. And when we place the cell in distilled water, pure water, this is a hypotonic solution. The water will enter into the cell and the cell will burst. So this is a diagram showing you the net movement of water in case the cell is placed in an hypotonic solution, in an hypertonic solution, or an isotonic solution. You can see here in an isotonic solution, equal amount of water entering and leaving the cell, so there is no net change in the cell volume. Here in this case, in a case of hypertonic solution, there is net outward movement of the water from the cell, which causes the cell to shrink. And in case of hypertonic solution, there is net inward movement of the water, which causes the cell to swell and can burst. The same thing shown here. Okay. Now I think you can guess this cell is placed in which type of solution. Yes, if you said isotonic, this is the correct answer. And this cell, as you can see here, the normal concave appearance, that normal indentation over here in the center is lost. The cell has lost its, and it is bulging. It is placed in which type of solution? It is hypertonic. And here the cells is speculated and the cell has shrinked. If you compare it with the normal cell, this cell has been placed in which type of solution? Yes, hypertonic. And osmosis, this process has much biomedical importance, because this osmosis has a role to play in many of the diseases like diabetes. When there is a loss of glucose, a lot of glucose going into the urine, and because all glucose is osmotically active, it takes a lot of water with it. It will lead to polyuria, a lot of urine production. And this principle of osmosis, the osmotic principle or the application of this principle is also seen in the dialysis, the hemodialysis and the peritoneal dialysis. The osmosis, this process is used to remove, this process helps to remove the waste products from the blood of a person whose kidneys are not functioning properly and not able to excrete the waste products. That is all for today. Thank you and Allah Hafiz.